Hello guys, in this video tutorial we'll be talking about a real-time PCR as well as we'll be talking about the quantitative PCR, which is also about the real-time application. Now we'll be seeing the difference of real-time PCR from the conventional PCR, because this real-time PCR is extremely similar like that normal PCR, but it is varying in a little way. What the name suggests is real-time, but actually the conventional PCR we have already discussed is called the endpoint or endpoint type of PCR. The endpoint type of PCR means those kind of PCR is giving us the information once the whole process is done, so we get the answer at the end. But in case of this real-time PCR, what we are looking, in case of this real-time PCR, we get the results during the PCR process, or as the PCR process is going on we are getting our results. So that's why this is varying. Now this real-time PCR is an identical to the standard PCR, except that the progress in the reaction is monitored by a camera, not actually by a camera, by different type of a detector, we must say. Now the detector is detecting the expression or the indication, right? The indication is coming from different dyes. Most of the time the indication is coming for fluorophores, so we get fluorescence, so we need to measure fluorescence. So we need that fluorescence detector for measuring the fluorescence that we are obtaining during the process is going on. Now hence the number of so what happens actually, the number of gene copies increases during the reaction, so as we know the fluorescence will also increase. Now in this picture you can see at the very beginning, in this vial we are having very little fluorescence. That means we are having very little PCR amplified product. Here we are having moderate one, and here we are having the largest or highest concentration of the PCR amplified product, right? So this is the system. So again, PCF tube in thermocycler, but it is attached via the detector system. We add the detector molecules with fluorophores there in very interesting techniques. We are going to study the different dye attachment techniques, but here this system is attached to spectrophotometer, which will measure and detect the fluorescence that are coming in, and by detecting the fluorescence we can tell whether the product or gene product is amplified properly or not. So what we can do here, we can actually quantify the amount of gene which is getting amplified. So the quantification can be obtained. So another type of real-time PCR is called the qPCR or quantitative PCR. We are going to see both of them together. So you'll blend this term real-time PCR and qPCR. So qPCR is a part of real-time PCR application, and it is vastly utilized to understand the concept of mRNA inside the cell, and also the concept the concentration of some amount of proteins inside the cell. Okay, and this PCR are important because they are having sensitivity 5 orders of magnitude better than any kind of blotting. Even the best type of blotting, which is the western blotting, we know that it can sense almost 1 nanogram of protein, but this kind of real-time quantitative PCR can sense uh, more than 5 times that of that western blotting procedure. Now the application of qPCR are found in many regions, so in gene therapy, and also expression assays, drug response analysis, mutation analysis, expression systems development, GMO analysis, and all these things. Now this qPCR are mostly utilized in the different GMO detection, single nucleotide polymorphism or SNP genotyping detection, as also the detection of pathogens. So it can be utilized as a diagnosis tool for diagnosing HIV diseases like HIV, cystic fibrosis, sickle cell anemia, thalassemia, phenylketonuria, and so on. We can also utilize this kind of qPCR processes to understand to get the concentration of maternal DNA in the amniotic fluid and all these things, right? So what is the basic procedure? We are having our mRNA. We reverse transcribe this mRNA into cDNA, then we do the quantitative PCR of the cDNA to get the quantification of the gene, and also a quantification of the gene expression, because the expression of gene means it's from DNA to the RNA, and from RNA to translated into the proteins, right? So we get the quantification of gene expression by studying any gene, or by studying the cDNA from mRNA utilizing the qPCR processes. Now, qPCR detection can be done utilizing instruments because it cannot be the simple conventional PCR instruments. There must be the normal thermocycler which will carry out the basic processes of PCR. That means first is the denaturation at 94 degrees Celsius temperature, then annealing at 53 degrees Celsius temperature, and then again the extension or elongation at 72 degrees Celsius temperature. So for this temperature variation, we must have the thermocycler, but the whole system is attached to thermocycler, so we must attach a spectrophotometer which can detect the fluorescence, right? So you need to have that. So here we are having that spectrophotometer. Here you can see, so this is so let us see the large picture. Yes, now what we are seeing here, we are having the thermocycler at the bottom, which is a major part of this instrument, and then we are having sample holders and tubes. Then we are having filters of 4 color detection of emission, because we are looking at the fluorescence, right? So for that we have 4 different colors here: red, purple, green, and yellow. So 4 color, and also software attachment there, and also it is the whole program is wired into the monitor, right? We must have the CCD array and also dichroic mirror for so you know that dichroic mirror and this 4 colored wheels and all these things are typical characteristics of a fluorescence detector machine. If you don't know how the fluorescence detection occurs and happens, you must go in my YouTube channel and you can find videos on fluorescence microscopy where I have talked in detail about this dichroic mirror and this filter wheels and all these things. I'm not going to talk about this anymore, but what is going on here, there is a procedure, and this whole system is arranged to get the detection of the fluorescence, right, which is coming out from the fluorophore which was added into the sample tubes during the PCR process. Now, what are the type of sample probes that are prepared for detection of the fluorophore? Now, there are 2 different types of detection probe. One is a specific, one other is non-specific. The non-specific type detection can be done utilizing the probe called SYBR green, or it is also called cyber green. The cyber green is a very common and important one. And in specific detection, we are having Tachman probe as well as molecular beacon. And primer based detection, we are having scorpion primer. So we are going to talk about this 4 in details: by cyber green, Tachman, molecular beacon, and scorpion. But molecular beacon and scorpion are pretty much similar in their functionality, so we'll be talking them together in blending. But cyber green and Tachman probe will are completely different type of detection. We are going to see them later. Okay, so we utilize these probes, but the actual function which is common to all these probes are that these probes are attached to a molecule which is fluorophore, so they can emit photons. So if we get those photons, there are detector system as we have seen earlier. So detectors can catch those fluorophores and can detect those fluorophores that are coming out from the PCR tube, and it can give us the quantification of what is going on inside the tube, right? So here are the different processes: cyber green probe mediated process, Tachman and beacon mediated process. We can see cyber green is a probe where you are seeing only one type of probe. On the other hand, in case of this Tachman probe, we are having 2 different things are attached with each other. One is called R, another one is called Q. One is Q means quencher and R means it is giving us the results. And these molecular beacons are little bit looks like a beacons, that's why it's called molecular beacons. And it is much more look like the stem loop structure, right? So there are varying in structure and also varying in their property. We are going to see the process in detail. Let's first talk about the cyber green. Now the cyber green is a DNA binding dye. It's simple, it's a DNA binding dye, and it binds to minor group, and only it binds to the double stranded DNA. Now this is the most important concept about the cyber green, because it can only binds to double stranded DNA and minor group of the double stranded DNA. So you can see the binding of cyber green here. It is a green colored dye as the name suggests, but it can only bind to double stranded DNA. So during the process of PCR, when you have denatured both the strands of the DNA, it cannot bind to the single strand. It can bind to single strand, but the binding is not that much tight. So the fluorescence that it will give us when it binds to the single stranded DNA is very, very low. But when it binds to the double stranded DNA, when the PCR is done, when the amplification is done properly, it can give us a strong signal of fluorescence. So what we can get during the beginning of the fluorescence, during the beginning of the PCR reaction, when everything is just denatured, we get very less cyber green activity. Then when it reaches the annealing and then finally elongation process, we get pretty higher concentration of this, pretty higher uh, power of detection by this cyber green. And by looking at this detection of fluorescence coming out or emitting out from the cyber green, we can actually tell the PCR product is amplifying with time, right? So say this is the process. So at the very beginning where the denaturation occurs, cyber greens are in the mixture, but they are having very less affinity towards the single stranded DNA, so very low amount of uh, fluorescence that is coming out. But when annealing is done, a slightly higher amount of fluorescence is coming because the hybridized content of primer with this our gene of interest or the template DNA is giving us a double stranded segment, and cyber greens are started to bind with them and giving us a little bit more uh, fluorescence. Then when the extension is done properly and we produced two separate gene of our interest clearly, then all the DNAs in the solution start to become double stranded and cyber greens start to attach to them, and the concentration, this attachment giving us higher concentration or higher magnitude of fluorescence, right? So by looking at or measuring the fluorescence, we can tell the PCR amplification is done properly. But again, this process is unspecific because the cyber green can bind unspecifically to the different region of the DNA, but it binds to the minor group and double stranded DNA sequence, that is for sure. Second process is a Tachman process. So the dye we utilize is a Tachman dye. Now Tachman dye is, you can see, it is consisting of two different regions. One is a R, another one is a Q. R is called the reporter dye, Q is called the quencher dye. Now here the quencher is red, reporter is green. Now what will happen when they are present in very close proximity? The reporter dye is eager to emit photons. Try to understand the concept. The reporter dye is eager to emit photons, so it is eager to emit photons in normal situation all the time. But what happens when this reporter dye is in close proximity with quencher dye? It will block the activity of reporter dye. It will block the reporter dye to emit any photons. That's why it is called quencher, right? It will block its activity of reporter dye. As a result, no emission is done, no photon emission is done, no fluorescence is measured when they are in close proximity like that. They are in close proximity when they are freely distributed in the solution of PCR mix. But when the elongation process is done properly, for example, say when they are ligated to our desired gene segment, again they are in close proximity because this Tachman chain is very short nucleotide sequence, so they are in very close proximity, and the quencher dye is quenching the activity of reporter dye in normal situation. But what will happen when you start the extension of the primer utilizing the Taq polymerase, which is given here in this purple colored enzyme? So this is the Taq polymerase. So when the Taq polymerase start the elongation process, it will eventually going to cleave the reporter segment. Now as it is cleaving the reporter segment and reporter dye is free now, our quencher is totally different. Reporter dye is free. Now as there is no quencher dye right behind the reporter dye to block its activity, so reporter dye can easily emit fluorescence, right? So reporter dye start emitting photons, so this fluorescence can easily be measured, and by looking at that we can tell that our PCR reaction is come to an end, right? So but this process is only done once after the Taq polymerase starts its action, so it's called the Tachman polymerase, right? Or Tachman process. Sorry, I should have discussed this advantage and disadvantage of cyber green before. Now the advantages and disadvantages of cyber green that we have just talked about on the previous slide is that advantages is that it is simple to design, just need to find a good specific primer as a target. It is sensitive and also it is inexpensive, so it's simple to design, sensitive and inexpensive. These are the positive advantages. But the disadvantages of cyber green is that it is unspecific. CyBR detects all double-stranded DNA. So if PCR is poorly designed, or primer dimer can also happen. So if primers are self-ligated with each other instead of primer DNA template, green is also going to fit into these primers and giving us the signal, right? Because it cannot distinguish whether the two strands are attached, they are primers or they are primer and template. They are unspecific, right? For that reason it can give us false measurements sometimes. It cannot multiplex a cyber green in quantitative PCR analysis. So you cannot use this in quantitative PCR because it can give us the wrong results due to because of it cannot distinguish between primer, dimer, or primer, template, dimer. Because in most of the case we want the result, or we want the fluorescence coming from the primer and template dimer, but not the primer dimer, right? Now we have seen the Tachman process. Now what is the Tachman chemistry actually? If we look at here in this picture, we can find it here. R is dark or quenched due to Q. We have seen it in this case. Now this is the Tach polymerase. It is acting like a chewing agent here. So it will come and it will start chewing. It will cut the reporter part out, reporter dye out. So it will start giving us uh, the fluorescence, right? So we can detect it. So this is in a more detailed process, and how this process is done. So at the very beginning, Tachman probe hybridizes to denature the DNA, and reporter fluorescence is quenched due to the proximity of the quencher, right? So this is the very beginning. The primers are sit and bound. Second thing is that uh, lengthening of the strands, as it starts to lengthen by the Tach polymerase. So let me draw, say this is the Tach polymerase. It will sit and start elongating the primer, right? As it is start elongating the primer, it displaces this Tachman dye, a Tachman probe, from the 5' end, and at the 5' end of the Tachman probe we are having the R, or reporter part, or reporter dye region, right? As it is start to displace this 5' end, and it will start cleaving this part out. So sooner what we end up with, the R, or reporter dye, cleaved out. As soon as the reporter dye is cleaved out, it is having less proximity from the quencher, it is having less activity from the quencher, so that it can give us the fluorescence, which is depicted here by this red colored uh, thing outside, red coat outside, right? So hydrolyzed Tachman probe eventually dissociates, and as the extension is complete, it will start giving us the signal, and we can measure the signal, right? Sorry, we have already discussed about the advantage and disadvantage of cyber green, so we don't need to discuss it here. Sorry. Okay. It shouldn't be cyber green. Instead, it should be Tachman polymerase. Sorry. So the advantages and disadvantages of Tachman polymerase. Not polymerase, actually Tachman probe. So this should be the advantages and disadvantages of Tachman. Sorry for the handwriting, bad handwriting. Now the advantage is that this Tachman probe is very specific, because it combines specificity of primers and specificity of the Tachman probe, right? Because both are interacting with each other in very huge specificity. It can multiplex qPCR assays to simultaneously amplify and detect different type of sequences. How we are going to say it later, because we can utilize several different type of Tachman probe altogether in the multiplex PCR. Remember, we have discussed about the multiplex PCR, which is a kind of PCR where we are amplifying several different gene sequence of our interest at the same time in a same single mix, right? We can use several different Tachman probe along with different types of reporter dyes, different color reporter dyes. So say this is one reporter dye color is say red, and this is another, it is having the reporter color green. So we can have different varieties of Tachman probe we can utilize in multiplex qPCR or quantitative PCR. Some disadvantages may also there. More difficult to design because of need of efficient amplification. Cybergreen was very easier to design because they are unspecific, non-specific. They can be designed just like that, very simple, easy to find. But they are complicated to design because we need to design in such a way that they can hybridize with our gene of interest, or the region of our gene of interest, and the binding should be efficient. so that it cannot be dissociated or cannot be hydrolyzed with pretty easily. And it is more difficult to design because the Tachman probes do not quench efficiently, right? This is also part of that. So it does not quench efficiently. If it is does not quench efficiently, the large background fluorescent and lower signal to noise ratio is there. As a result, it will become extremely hard to distinguish the actual fluorescence that is coming out from our probe because of the background noise, because this is less hard to quench, and if it is getting quenched, the reporter start giving us this response from outside. If anyhow uh, before the binding into the DNA it breaks from here, it can give us separate parts. For example, one part, say this is one part, another part is this. So as soon as it is separated like that, it will start giving us response, it will start giving us fluorescence, right? Suppose this Tachman probe breaks down from the bottom before even added to the DNA segment, so it will give us this kind of fluorescence, right? So we think that so in this case of results, we can think that this is coming out from our PCR product, but this is not actually coming out from our PCR product. Instead, it is coming out from the spontaneous hydrolysis. So this kind of spontaneous hydrolysis of Tachman probe can give us bad results and higher background noise. So we need to cancel this background noise here for to get a perfect result. Okay, for some target sequences, AT-rich sequence make probe design difficult. Okay, these are also several difficulties. So difficulties are difficult to design, and also spontaneous hydrolysis can create problem. Rest of the things are fine, but they are very specific, right? Now let us talk about the molecular Bacon probes. As I have told you before, these are the third type, molecular Bacon. Now this kind of probes, what are the structural conformation of them? They are looking like a stem loop structure. So this is the stem and this is the loop. Now this to have this structure is very important, because again in this case also we are seeing two different parts. One is the reporter die, another one is the quencher die. So reporter and quencher die are placed in very close proximity due to this kind of stem loop holding. So what we know from the previous discussion, that this quencher die will block this reporter die's activity to emit any photons, right? So it won't give us any photon in normal situation when it is that. But another important feature of the Bacon probe is that Bacon probe is having the stem loop structure along with the binding of 5' reporter and 3' quencher. This quencher is indeed bind with another sequence which is called the blocker or the PCR blocker sequence. This is also important. This is called the PCR blocker sequence. Now this utilizing this PCR blocker sequence, it can actually hold on to the 5' end of the primer that we use for the PCR analysis process, right? So here we can see at the very beginning of the reaction, when it is in its stem loop form, with the help of this blocker region, it will bind to the 5' part of the primer. So it will bind like this just on. Then what will happen, as the PCR begins, denaturation happens, and as a result of this high temperature, it will break down the hydrogen bonds that are present between this stem structure of this Bacon molecule and Bacon probe. So what will happen, it will get linearized. So as it is getting linearized, it can hybridize with the template strand of our interest instead of the intramolecular hybridization. So previously it is hybridized with itself, but now as a result of this heating at 94 degrees Celsius temperature for the denaturation in PCR, it is getting linearized and it can hybridize with the DNA strand or the newly synthesized template DNA strand. So what will happen now, due to this hybridization, this quencher and reporter are in less proximity, so they are distantly apart now. So now this reporter can emit photons. So reporter can emit photons and it will start providing us the signal of fluorescence. So we can measure the signal of fluorescence due to this hybridization of this Bacon probes with the newly synthesized DNA strands. Okay, so that's how we start getting this thing. So what is, if you look at the graph of fluorescence in y-axis and temperature in x-axis, when we start our PCR reaction without any kind of Bacon hybrid or Bacon probe, what will happen, as we are rising the temperature, we are getting here say less, very very less, no fluorescence at all, because there is no Bacon hybridized with the template. But as we are increasing the temperature, it is getting opened, and we will start to emerge a slightly little fluorescence. But when we give the Bacon target hybrid at the very beginning, so you can see here the Bacon is in stem loop structure, so it will give us very very less fluorescence due to this quencher activity onto the R, because the quencher and reporter are in very very close proximity here. So it, as we are increasing the temperature, it will give us some fluorescence due to this reporter activity, because quencher is not that much efficient to block the whole activity of reporter even if they are in close proximity. So they will give us a very very thin line of very very pure line of fluorescence when they are in close proximity. But when they are in distance, when they are in distant separate and distantly apart, in those conditions they can give us a high fluorescence value. As we increase in the temperature, they start giving us so as we increasing the temperature, they again start giving us. But as we increase the temperature again, in this case, remember, because they are already hybridized, so they start giving us very very high fluorescence. But again we are heating them, so they are getting separated, and there should be another chance of self ligating with itself. So in those cases, heating this Bacon target hybrid for another time can give us a less fluorescence, because it will increase the chance of self ligating of this Bacon and produce the stem loop structures again, which will give us a small fluorescence, right? So in both the cases we can see how the fluorescence is changing. For the Bacon without the target, as we increase the temperature, it is going towards the self ligating portion towards the Bacon with target, and it will give a rise in the fluorescence. But when we are looking at hitting at the Bacon target hybrid, there is a tendency to shift back towards its original conformation, which will eventually lower the fluorescence down, right? So this is a clear cut understanding, and this understanding is pretty important. Now in this picture we can see, utilizing this Bacon as probe, we can get this fluorescence detection, beautiful fluorescence images inside the cell, and looking at the number of mRNA or complementary DNA that are being amplified via the PCR process. Now let us talk about the scorpion primers. As I have told you before, they are resembling very very similar in their functionality with with this Bacon probes also, because the scorpion primers, as you can see here, they are also having the stem loop structure with this blocker segment. So as we heat them to denature, so they will become denatured, and due to this denaturation they are distantly apart now, so this F part start giving us the fluorescence. And as we start to cool them down, they are having a tendency of again resealing with each other to find this stem loop structure again. So the fluorescence activity, which was raised which was previously uh, higher, now start to go down. Then what will happen, we start the extension process. When you start the extension process, again the denaturation is done, and again the uh, this stem loop structure is broken down. As a result of stem loop structure broken down, again the F is distantly apart from this Q or quencher, so it will start giving us fluorescence. So again there is a rise in fluorescence, again there should be a fall in fluorescence due to the cooling, right? So in this way we can find this up and down graph. If we draw the graph for this scorpion primers, whenever they are ligated, the temperature is cooling down, and we give the enough cool temperature for them to religate. They will religate with itself to make this their native structure, and in those conditions the concentration or the signal for fluorescence is going down. But when again we denature them, again F part is coming out, and it is giving us the fluorescence signal, so fluorescence will start up. So this is the process of scorpion primer fluorescence detection. Now we can detect we can use actually this TACMAN and also this this stem loop structure of scorpions, and also the beacon probes in case of the multiplex PCR, because we can utilize several different colors of that. So green color and purple color is taken in both the case. So green color is R1 or reporter 1, this purple color is R2 reporter 2. This R1 is targeted for a gene G1 and this R2 is targeted for the gene G2. So we are, this gene 1 and gene 2 are our interest in this case of research. So in the multiplex PCR we want to study the expression of gene 1 and gene 2 utilizing two different sets of beacon probes. Now they can bind and giving us the fluorescence, two different types of fluorescence, one is green, another one is purple. We can detect it from here, and by looking at this graph, cycle number in x-axis, fluorescence in y-axis, we can actually tell uh, the concentration of G1 and G2 inside this PCR mix. Okay, so this is a pretty good to understand the comparison of different mRNAs and the expression of the mRNAs inside the cell, right? Now let us talk about the most important application of this real-time PCR, which is called the quantitative PCR. Now quantitative PCR, as I have already told you, that it deals with the expression of the mRNA. So what we can do, we make a sample by the DNA extraction or RNA extraction, whatever. By extracting DNA or RNA, we get our sample. Then if we get DNA, we can directly utilize it for the qPCR process and we analyze the data. But if utilizing the RNA, then we need to make complementary DNA, because we can only amplify DNA segments utilizing PCR. We have already talked it before. So we need to make a reverse transcription of this RNA to make the cDNA, and also we must treat it with RNAs to degrade the strands and the process of getting the cDNA, and then we can utilize that cDNA for this quantitative PCR process. But the amount of cDNA that is produced must be corresponding, and it will vary with the amount of, it is proportional with the amount of RNA which was present in the extract. So by analyzing the data we can compare our results. Now before understanding the qPCR process, we must understand a very basic of this real-time PCR product. Now the real-time PCR is having three different states or three different phases. What are the phases? First phase is called the exponential phase. Now what do we mean by exponential phase? At the very beginning of the reaction, we are having plenty of template, plenty of primers and all these things inside our reaction mix. So the reaction will start pretty rapidly and it is giving us product exponentially. After certain time, when it reaches a particular concentration of our desired gene product, in those conditions the gene product is continuously getting increased, but this phase is called the linear phase. So this is the second phase or linear phase. Now the third phase is that in that condition, when after certain times, when after 30 cycles, remember we have discussed it earlier, after almost 30 cycles of PCR process is done, what happens, there are limitations because the probes and primers and also the DNA templates start to be less, and in those conditions the PCR process is start to halt. So what we get, a very linearized value like that. This stage is called the plateau stage. So there are three different phases of a PCR process. Okay, and you need to understand these three phases pretty importantly. And another thing you must know is this value or CT value, which is called cycle threshold method. Now what is that? Before that I must tell you another important thing is that, so during the exponential phase of PCR, the amount of PCR product is proportional with the amount of starting template. This is a very important assumption. During the exponential phase, amount of PCR product is proportional to the amount of starting template. So the PCR product, if we get if we start with high amount of template, we end up with high amount of PCR product. That is pretty logical, we all know that. Now that was initially added, it is very important. Therefore the exponential phase of PCR is analyzed to quantitate the initial template concentration. Suppose we don't know the initial concentration of the mRNA we are dealing with, right? The initial concentration of mRNA we are dealing with. in this case in the PCR we are not taking mRNA, we are taking cDNA, but we know that the cDNA concentration is exactly directly proportional to the concentration of mRNA inside the cell, right? So we don't know the initial concentration. So after certain time period, if we calculate the value of the concentration of our end products, of our amplified products, it should be proportional with the starting product, so we can get back to the starting product. And if we compare, suppose we comparing three different type of samples, so here is the sample 1, sample 2 and sample 3. We don't know the initial concentration of these three mRNAs inside the cell, but after running this PCR process, what we get is that for example, say not this, say this one, sample 1 so this is the sample 2, this is sample 3. Now we don't know the initial concentration of this mRNA, which is sample 2, but what we know, we also run the PCR for sample 1 and sample 3, the concentration, the initial concentration of which we know. Now what we get from it, we get the amplified product of it. Now we get the amplified product of both of them, 1, 2 and 3, all of them. And what we can conclude from that, the amplified product of 1, from the amplified product of 1 we can look back the starting concentration which is known, and again from the amplified product of 3 we can look back to the starting concentration which is also known. And using these two as a standard measurement, we can measure the starting concentration of unknown product 2, because the end concentration is known by us. The end concentration of 1, 2 and 3, all of them are known to us after the amplification process. And how we know the end point or end concentration of the products? Utilizing several different probes, utilizing the probes we have discussed about. So this is the magic of quantitative PCR. This is the magic of real-time PCR, because we are measuring the concentration of the amplified product of our interest directly via the measurement of fluorescence. So you can measure this all, and among these three, the starting concentration is also known for 1 and 3. So we can utilize the starting concentration of 1 and 3 and also the end concentration of 1 and 3 to make a standard curve. Utilizing the standard graph, we can plot the unknown starting concentration of the sample 2, of which we know the end concentration, right? So from this end concentration we can track back the starting concentration of this sample 2. So from this starting concentration we can tell that this mRNA is transcribed, the expression of the mRNA can be identified. So that's why it is called the quantitative PCR. So we are quantitating the amount of mRNA, or we can amount of gene, any kind of gene at the very initial stage, right? This is very very important. But what is another important part? So the initial template, so the commonly used method is called the cycle threshold method for that. Now what we mean by this CT or cycle threshold method? The CT value is a value at which what we can say during the exponential phase, after a certain time, what happens, the concentration of the product is pretty higher, so that we can designate that, we can separate them out from the background noise. So that is called the cycle threshold or CT. It means the concentration of our desired sample mm, fairly measurable from the background noise, or can be separatable from the background noise. Now at the very beginning when we started the process, as we can see, all of these things are creating mess, so there are a lot of background noise and the concentration that we are getting is pretty less, so we cannot conclude by looking at here. So after certain time, when you can see this orange line which is called the threshold, after certain period of time, it start providing us, when all of this process start to enter into the exponential phase, it start providing us huge amount of fluorescence concentration. And by looking at this fluorescence, we can actually detect and we can tell that this fluorescence signal is coming out, which is much more higher from the background noise, so we can actually detect it. And this particular line which is called the cycle threshold, or above which we must So the cycle threshold is giving us or telling us the important insight that whatever we need to quantitate, whatever measurements we need to take, we must take it above the cycle threshold, not below the cycle threshold, because below the cycle threshold there are a lot of noise, so we don't get appropriate result. So to get an appropriate result, we must take the detection above the cycle threshold. So whatever we are getting in cycle threshold or above the cycle threshold, we'll take that out, and during the process we are measuring the fluorescence activity. Remember, another very major difference between this real-time PCR and conventional PCR is that we are measuring the product at the end point in case of conventional PCR, but in case of real-time PCR we are measuring the product, or we are measuring the uh, signal which is coming out from the product after each cycle, during the process, when the process is getting progressed, right? So that is the difference. So it is giving us another important insight, that if we are quantitating it at the end, it can't give us a sufficient result, because it will reach the stationary phase. So we must carry our all the discussions and results in their exponential phase, because this is the appropriate phase for knowing uh, the exact dynamics of this PCR. And also in this exponential phase we can tell this fact that the amount of PCR product is proportional with the amount of starting template, right? So we can get these results, and by looking at it we can tell what is the unknown concentration of product. So that's why this particular graph is really important. Cycle number in x-axis and fluorescence in the y-axis, or we can place time in the uh, x-axis, because cycle number and time can be placed similarly there. Okay, so you can see after 30 cycle plateau value is reached in this case. So whatever type of detection we are going through, the detection is from 15 cycle to 30 cycle. This is a very important part. This is the very important time where the products are uh, exponentially increasing. Okay, now the applications of real-time PCR and also their advantages. The advantages are, this is very sensitive and very complicated also, but very sensitive. uh, we can quantitate the gene sequence by comparing the pattern of known sequences, as we have seen earlier, and they are very specific also. So the measurement that we are getting it very sensitive as well as very specific. These are the major advantages of a PCR. So any kind of quantification, if you are trying to run any quantitative analysis, the most important part is that you need to be very specific and sensitive, and both the things are fulfilled by the real-time PCR. So it's a bonanza for us. And the applications, we can apply it for the detection of genomic DNA or viral DNA. So as we can detect the genomic DNA or viral DNA, it can be a very valuable diagnostic tool for us. Second thing is that the gene expression can be measured, so we can quantitate the gene expression pretty easily. So after extraction of the total RNA and preparation of the cDNA from this RNA via the reverse transcriptase, we can measure the gene expression of those RNAs, as we have seen in the previous case how they measure the RNA content, right? And also the processes can be extended, and the analysis is simple and can more easily be extended, because all the process that we have seen, the graph analysis and everything, is nowadays done by softwares. So you just put commands and sit onto the chair and look at the monitor screen, and they will be telling you all, right? So in a sense, in all that, real-time PCR is really a bless to us. So it's very good, and I hope this video is helping you to understand real-time PCR. Thank you.